Good day, we are Group 6 from Kuliah 6 and me, Foo Tongli with Shivanaysan and also Natasha. We are going to present on the topic of evolution of animals. So let's proceed with our presentation today. The first one is the characteristics of animals. So there are similarities in the characteristic of the animals as well as differences. So let us see on the similarities. First, the animals, they are multicellular, which means they have more than one cells in their body. So just like plants and also fungi, they are all multicellular organisms. They are made up of multicellular eukaryotes. And the next one, animal cells is lack of plant cell, in which we know that lack of cell wall, because cell walls is only present in the plant cell. And then all of the animals, they are heterotroph, which means that they acquire their nutrition from external sources but not producing by themselves through process like photosynthesis. So they are also mobile because they have well-developed nerve and also muscles to help them to move around. Most of the animals, they reproduce sexually. And the last similarities of the animals is that they undergo a series of developmental stages to produce an organism with specialized tissues. Okay, so let us see on the differences between the animal. So animal, they have vertebrates and also invertebrates. For vertebrates, it means that the animal it have spinal cord. For invertebrates, means that they don't have spinal cord, which is known as the backbone. Okay, and the last differences is that they are categorized into haploid, diploid, and also haplodiploid. So haploid is the half of diploid, which is 2n, so haploid is n. And haplodiploid means that they are consist of 2n and 2n. So it is actually a sex determination system in which the haploid male is developed from the unfertilized eggs, while the diploid female is developed from fertilized eggs. Okay, so now let's move on to the next slide. Okay, now you can see here the next slide here show that the colonial flagellate hypothesis. So this hypothesis stated that the animals are descended from an ancestor that resembles a hollow spherical colony of flagellated cells. So one of example is the choana flagellate. So this cell is one last kind of single cell ancestor of animals. So one single flagellate cell, you can see there is a flagellum there for their mobility to move around. So the motile flagellates will form an aggregate of flagellated cell, and then this aggregation of the colony of cell will then form a hollow sphere as shown here, and then they will undergo specialization to have specific function. For example, for is shown here, reproduction, so it is developed into reproductive cell. So it will then enfolds and creates tissue, and this is how our tissue layers is formed. Moving on to the next slide, this is the phylogenetic trees of animals. So this phylogenetic trees actually represents the evolution of animals. So these are all they are all categorized as the animals. So this is based on the combination of morphological characters of living and fossil organism, development of homologies, and also the molecular characteristic. So just for your knowledge, the morphological and also molecular data is actually the rRNA sequencings in a certain organism. So as you can see from these phylogenetic trees here, they are starting from the same ancestor, choana flagellate ancestor, and then they will branch into a lot of different characters and then group into different groups based on their characteristic. For example, the first one, either you have tissue layer or not, and then you have bilateral symmetries or not. The next one is that deuterostome development. You will go to the upper side, and if you if the animals is protostome development, so it will go to the down part, as well as the molting of cuticle and also the spiral cleavage. So you can see in this tree here, they are actually the two red dots, right? So actually these two red dots, this is to represent the uncertainty in the researchment in the ctenophores at the down part there, and also the spirilla groups, because there are still uncertainty in the researchment of the relationship in this organism, so they put the red dots there. And the more closely related two organisms are, the more similar their DNA sequence. So let's have a look at these phylogenetic trees. Okay, so now that you have understand the phylogenetic trees of animals, let us proceed and learn another thing about animals. Evolution of animals is that there are two types of symmetry here. So before we talk about the type of symmetry, we must know that there is actually symmetry and also asymmetry of a certain body structure. So for asymmetry, it means that body structure, body shape do not have a certain pattern. For symmetry, it is the opposite, they have a certain pattern for that. So okay, there are actually two types of symmetry for animals, which is the radial symmetry and the bilateral symmetry. So let us look at the radial symmetry. The radial symmetry is actually that the body of the animal, they are organized radially, circularly, such that when you give a longitudinal cut through their central point of their body, it will actually produce two identical halves, which means like cut into half, which is actually the same shape. So for example, take a look at this coral. It is actually radial symmetry animals, and these type of animals, they are either mobile or immobile, so it depends. And this symmetry will allow the organism to extend in all directions from one center. Okay, the next symmetry is the bilateral symmetry. So for this type of animals having bilateral symmetry, they will have definite left and right half, just like human. Okay, they only have having a single cut, longitudinal cut down, where cut down the center line of animal, so it will produce two equal half of the animal, just like having a look at this is a bit that you cut into half, it is actually symmetrical. So they have the definite anterior and posterior ends in their body. Anterior is like the front part, posterior is the behind part there. So actually this is accompanied, this animal, they are accompanied by self-fertilization, which is the location of a brain in specialized sensory organs at the anterior end. This is actually giving them the ability to engage in directed movement as their wish. They want to move to their stimuli, they move to find their mate, find their food. So I will give you another two examples of this symmetry. For example, the radial symmetry is jellyfish, and the bilateral symmetry is turtle. Okay, so that's all for my part. I will pass to Shivanaysan to talk about the animal kingdom. Thank you.
Now, let's dive a little bit deeper into the animal kingdom, domain Eukarya, kingdom Animalia. The animal kingdom, as we know it, is separated into two groups, invertebrates and vertebrates. Invertebrates are animals that do not have a backbone, and vertebrates are animals that have a backbone. Let's take a closer look into the invertebrates. There are 5 groups of invertebrates that we should know, and starting with the simple invertebrates. There are 3 kinds of simple invertebrates, which are sponges, ctenophores such as comb jellies, and cnidarians such as hydra, jellyfish, corals, and sea anemones. Next group is called Protosomia platyzoa, which are made out of rotifers and flatworms such as planarians, tapeworms, and flukes. Next up, we have Protostomia edisosoma, which are made out of roundworms such as acaris, pinworms, and hookworms, and also arthropods such as crustaceans, spiders, centipedes, and millipedes. The fourth group of invertebrates that we should know is called Protostomia lophotrochozoa, which consists of lophophorates such as lamb shells and bryozoa, annelids such as polychaetes, earthworms and leeches, and also mollusks such as chitons, snails, clams, and squids. And the final invertebrate group that we should always remember is Deuteriostomia, which is made out of echinoderms such as sea stars, sea urchins, sand dollars, and sea cucumbers, as well as chordates such as tunicates and lanceolets. Next up, let's learn about the vertebrates. There are 5 different groups of vertebrates that we should know, starting with fishes, amphibians, reptiles, birds, and finally, mammals.
Hi guys, assalamualaikum and very good day. My name is Natasha. So today I would like to continue our presentation with the next slide that is about the embryonic development between the uterostome and also the protostomes. So move on to the next slide. This is in the terms of the trypoblastic animals. So as you guys can see, for the protostomes, the mouth actually appears developed prior before anus, while for the deuterostomes, the anus actually develops prior to the mouth. And the example for the protostome is the roundworms and also the arthropods. And while for the deuterostomes, the examples are ourselves, that is humans, and also the vertebrates such as the snake. And the next slide is in the form of cleavage. So for the protostomes, the daughter cells sit in groove formed by the previous cleavage, and for the deuterostomes, the daughter cells sit right on the top of the previous cells. So as you guys can see from the diagram above, you guys can see that the cell, in the top view of the protostomes, it is more like not in order arrangement compared to the deuterostomes. The next one is about the blastula formation. So for the protostomes, the mouth appears at or near the blastopore, but for the deuterostome, the anus appears at or near the blastopore. So the position of mouth and also anus between the protostome and deuterostome are actually inverted, like they are opposite to each other. Like for the protostomes, the mouth came first before the anus, but for the deuterostome, the anus comes first before the mouth. Next, I move on to the coelom development. So for the coelom development in protostomes, the mesoderm arises from cells located near the blastopore and split to form the coelom. While for the deuterostome, the pair of mesodermal pouches of the primitive guts enlarges, meets and fuses to form coelom. So based on the diagram, you guys can see that the mesoderm for the protostome, it is already separated from the cell, but while for the deuterostomes, the mesoderm is actually fused together with the cell, so they need to split before they form into the coelom at the next stage, while for the protostome, they are already separated at first. So I think that's all from my group, from us. So I hope that you guys can understand this topic very well, and good luck. Thank you for watching.